If we stored the entire DNA sequence of a single human cell in books, we would have 3,300 books, each with 1,000 pages, where every page contains 1,000 letters. Yes, a human genome, which has about 3 multiplied by 10 to the power of 9 base pairs, can produce such an enormous amount of data. In fact, an ambitious international scientific research project called the Human Genome Project, or HGP, set out to do just that, to determine the complete sequence of base pairs of DNA inside the human genome. More precisely, the HGP aimed to identify approximately 20,000 to 25,000 genes in human DNA. The average gene consists of 3,000 bases. However, gene sizes vary greatly. Did you know that dystrophin is the largest known human gene, which has 2.4 million bases? Now, if you add the number of base pairs in the human genome, the sequences of more than 3 billion chemical base pairs would have to be determined under the HGP. Therefore, by aiming to understand the genetic makeup of the human species, the HGP served an important purpose. It was important because it is a sequence of bases in our DNA that determine our genetic information. 99.9% of all nucleotide bases are exactly the same in people. Yet we differ from each other, and that is because our DNA sequences differ in certain places. Scientists have identified about 1.4 million locations with single-base DNA differences, also known as SNPs, or single nucleotide polymorphism. Knowledge about variations in the human genome can equip us with more ways to diagnose, treat, or even prevent disorders and ailments that affect human beings. Similarly, knowledge of the repetitive sequences in DNA shed a lot of light on chromosome structure and evolution. Repetitive sequences, also known as microsatellites, are those stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times, in the human genome. Also, they have no direct coding functions. In fact, less than 2% of genome codes for proteins, and for more than half the discovered genes, are still unknown. With the advent of advanced genetic engineering methods, it was now possible to isolate and clone a piece of DNA to determine its sequence. So even though the HGP was a mammoth project, it was a viable one. Since the HGP aimed to store the entire genome information in databases, it required high-speed computational devices for data storage and retrieval. In addition, the HGP required improved tools for data analysis. Thus, the HGP is also credited with contributing to the rapid development of a new branch of biology called bioinformatics, which involves the application of statistics and computer sciences to the field of molecular biology. The HGP was a 13-year project that began in 1990 and was completed in 2003. It was coordinated by the U.S. Department of Energy and the U.S. National Institute of Health. Did you know that initially it was James D. Watson, one of the co-discoverers of the double helix structure of DNA, who headed the project? However, in the initial years of the HGP, Wellcome Trust of the UK became a major partner, though more contributions arrived from countries such as Japan, France, Germany and China, to name a few. Although the HGP had aimed to understand the genetic makeup of the human species, it also focused on several other non-human species such as the bacterium E. coli, yeast, a free-living non-pathogenic nematode called Caenorhabditis elegans, a fruit fly called Drosophila, and the laboratory mouse. Learning about non-human DNA sequences can lead to a further understanding of their natural capacities and traits, which may offer solutions in various sectors such as health, agriculture, and energy production. Apart from transferring information and technologies to other sectors, the HGP also intended to address ethical, legal and social issues, or ELSI, that may have developed while working on the project. Therefore, though the HGP was a mammoth and hugely beneficial project, it required enormous effort, cost and innovation as well as caution to accomplish it successfully.